Hello, and welcome to our discussion of neurons and neurotransmitters. This will be our first topic under our neuroscience review. We're going to start off talking about the different levels of analysis we can think about when we talk about neuroscience and neuroanatomy. The lowest level is, of course, a molecular level of analysis in which people focus their attention on specific chemical substances and molecules on and in the brain. The next level of analysis would be cellular, and this would, of course, focus on neurons and support structures in the brain at the cellular level. We're going to focus quite a bit of detail on neurons and how they function here in just a moment. Neuroanatomy would be our next level of analysis in which we can examine the brain and nervous systems based on their structures. So we can look at specific anatomical markers in the brain and see how those structures might relate to various behaviors. And we're going to talk a little bit about that in other lectures. Neurophysiology is, of course, looking at various brain functions and where they might occur in the brain. It's a classic anatomy and physiology distinction. Anatomy is structure, physiology is function. We'll concern ourselves with both of these aspects of neuroscience at various times throughout the semester. So keep that in mind. Neuropsychology is basically looking at how individuals with some sort of brain malfunction or disorder or disease function, how to help them function better, and how we can understand the brain and its properties better through these patient populations. There's a great book, if any of you are interested in it, called The Man Who Mistook His Wife for a Hat by Oliver Sacks. It's a really great clinical assessment of various neuropsychology patients, and I highly recommend it. Neuroscientists tend to be less clinically focused and more research focused, and they're more focused on understanding structure and function and different cellular processes, molecular processes, and how they relate to cognition. And we're going to take a look at some research methodologies in another lecture. Give you a little preview. This is a couple of different views of a neuron, or actually a couple of neurons, in particular what's called the synapse or synapse. And this is really the lowest level at which we're going to look at how the brain functions. Everything that we are and do happens based on billions of these synapses firing all the time, and of course we have no awareness of that. So everything we are occurs at this level, which is kind of interesting to think about. But let's talk about neurons. Neurons are, of course, individual cells contained throughout the brain and throughout various parts of the body. We often talk about nerves. So, for example, we might talk about a nerve in your arm or severing a nerve. Those of you who go on to do neuroanatomy will have to learn the cranial nerves. These are bundles of neurons. We're going to talk about some of these at various times, but it's really not the level at which we're going to focus. We're more interested in how these neurons function and how drugs affect that functioning. Now, when we take nerves together and put them into a system, we have a nervous system, and we have a variety of them. We have a central nervous system, a peripheral nervous system, we have an autonomic nervous system, and a variety of sympathetic, non-sympathetic, all kinds of nervous systems. And we're going to talk about those later on. But we're going to start off talking about neurons. And again, these are individual cells that really do accomplish everything in the brain. And these are the various structures that you're going to need to know because they are critical in understanding how neurons function. The dendrites, the cell body, the axon, and at the very end of the axon is what are called the terminal buttons. This is a micrograph of a neuron. This would actually be a pyramidal cell from the hippocampus. You can actually see it looks like a little pyramid. If you look right here. So let's get into talking about the specifics of each of these individual parts of a neuron. The cell body is just like any other cell body from any other cell. It contains a cell nucleus, which is, of course, the metabolic center or power source of the cell. So a variety of things are going to occur in that cell body. Mostly it's just keeping the cell functioning. It's providing energy for the cell and replacing various substances throughout the neuron. We're going to talk a little bit about the cell nucleus and how it might be involved in creating some of the neurotransmitters we're going to talk about. The next most important structure for us to think about in the neuron is what's called the cellular membrane. And this is the outer covering of a neuron, and this is really important for understanding how neurons function. This is a semi-permeable membrane that encloses the entire neuron, and the structure of this membrane is critical to understanding neurotransmission. I've posted a couple of YouTube videos that describe this in more detail, so you'll want to take a look at those because they describe them a little bit differently. And certainly we want to make sure you understand this neurotransmission process. So here's the cellular membrane. It's basically a fatty layer or a lipid layer, which includes what are called ion channels. And these ion channels can be open or closed, and based on them opening or closing affects how many ions are inside the neuron and how many are outside the neuron. Based on that number of ions inside and outside the cell itself, this creates a charge because there is an electrical difference between the inside and the outside. Just like when you were in high school chemistry or middle school science and you learned how to make a battery through a variety of functions. Basically just like that, because there is a difference in the amount of ions across that membrane. So it creates an electrostatic charge. So the semi-permeable membrane that encloses the neuron, which is critical to neurotransmission, functions primarily through these ion channels that open and close to create an electrical charge. So while a neuron is just sitting around, it has what's called a resting membrane potential. So there's always some sort of difference between the inside and the outside of the neuron. Well, this dramatically changes when a neuron fires, what we call an action potential. So it's going to generate an electrical charge. It's going to travel down the length of the neuron, cause the release of neurotransmitters into the synapse. Those neurotransmitters will bind to the next neuron and then create an action potential in that neuron. And then that's essentially how everything functions, is through this handing off of signals through action potentials. To give you an idea of how this resting membrane potential functions, there are potassium and sodium ions inside and outside of the neuron. And we have electrostatic forces that are trying to force sodium ions in and potassium ions inside the membrane, inside the cell, pardon me. Inside the neuron, there is an excess of potassium ions, and through just the process of diffusion, if you remember the basic chemistry, when you add an ion to a solution, it tends to distribute itself because those ions repel one another, and so just through that process of diffusion, you end up with those ions distributed. So there's a force trying to push those potassium ions out. There's also a force trying to push potassium ions in and sodium ions into the neuron. We're going to come back to that resting membrane potential and how it affects the process of neurotransmission here in a moment. So the entire neuron is covered in this cellular membrane. Here at what we call the receiving end of the neuron are what are called dendrites. And this is where neurotransmitters will bind to open and close those ion channels to generate an action potential. So they're these short bushy fibers that branch out from the cell body, and these are associated with what we call the postsynaptic neuron. The synapse is just a space. It's simply a gap, and it's often called the synaptic cleft. And so when we talk about something being presynaptic or postsynaptic, we're talking about the neuron that's before the synapse or after the synapse. So the dendrites are associated with the postsynaptic neurons. Again, very important for understanding neurotransmission because neurotransmitters will bind to those dendrites and affect the process of neurotransmission. The axon then is a single long fiber that extends from the cell body down to the terminal buttons and then will end in the next synapse. So at any synapse, the terminal buttons will be up against the dendrites of the next neuron. So these dendrites up here would have terminal buttons associated with them, and maybe even several neurons converging on this one cell body and their dendrites. These terminal buttons then are going to be attached or nearby the dendrites of another neuron. Now an axon can be very short or very long. So for example, there are single axons that travel from your spinal column all the way down to your foot. So they can be several feet long. Keep that in mind because it's important for understanding how things like reflexes and muscular coordination, all of that functions through this process. So the single long fiber that extends from the cell body, and this conducts signals from the cell body to other parts of the nervous system, so that next neuron, and this is associated with the presynaptic neuron. So the single fiber and the terminal buttons are all part of the axon. So the terminal buttons, some people call them boutons, it's buttons in my world, are associated with the endings of the axon. And this is where neurotransmitters will be released into the synapse. And we're going to talk a lot about that process later on when we talk about neurotransmitters. Now notice that this axon is covered with these little nodules, and this is called myelin. This is what we would call a myelinated neuron. It's covered with this white matter, and in fact, when we talk about white matter versus gray matter in the brain, white matter are myelinated neurons, gray matter is neurons without myelin. So this covers up the axon, and what this functions to do is it supports the neuron, but most importantly, it speeds up the process of neurotransmission. The way a neurotransmission works is the dendrites signal the beginning of an action potential, which will be this electrical charge traveling down the axon. And what's going to happen is ion channels will open and close, sort of propagating along the length of this neuron. Well, what happens is because the cellular membrane is covered by myelin, the action potential is generated in these little spaces. And in fact, what happens is, and these are called the nodes of Ranvier, is the action potential jumps from one node to the other. So this greatly speeds up the process of neurotransmission. It's a really important process for understanding how neurons work. To give you an understanding of how important this is, multiple sclerosis, which is a neurodegenerative disease, occurs when the immune system of the patient, of a multiple sclerosis patient, starts attacking the myelin sheath as though it were something invasive, like a virus. And so what's happening is the person's immune system is actually degrading the myelin sheath. And what ends up happening is reductions in sensory and motor functioning. So this myelin sheath is responsible for what we call saltatory conduction, which is, in some dead language, a different word for jumping. So what happens is our electrical signal travels from node to node in these depolarized regions. This is critical for motor functioning, and again, multiple sclerosis is a demyelinating disease. So these are nodes of Ranvier, and our action potential jumps from one node to the other because these ion channels where the myelin is covering them can't open and close because they're covered. And so it gets propagated from one node to the other. So it's a very brief introduction to neurons and their structure. I want to talk a little bit about the process of synaptic transmission. And this is how neurons communicate with one another. And what happens is they relay information by releasing neurotransmitters. And what we refer to the space between two neurons as is the synapse, which we've talked about earlier. And that's why it's called synaptic transmission, because the information is relayed across the synapse. So the synapse is that narrow space between the terminal button of one neuron and the dendrite of another. Again, basic look at how this functions. Here we have a terminal button where neurotransmitters will be released into the synaptic cleft. Those will then bind to the receptor sites on the dendrite of the receiving neuron. And this is critical for understanding how drugs affect behavior because they tend to affect this process right here. Again, presynaptic neuron is the terminal button, postsynaptic neuron is the dendrite. Excuse me. An important part of understanding synaptic transmission is understanding neurotransmitters. And we talked about this already when we talked about pharmacodynamics. So these are chemicals that are released by the presynaptic neuron. They tend to then hang out for a bit and then either get metabolized or taken back into the presynaptic neuron. Most of the drugs we're going to talk about affect this process of neurotransmission by causing the release of more neurotransmitter, by blocking the reuptake of the neurotransmitter, by blocking the metabolism of the neurotransmitter, or by mimicking the action of the neurotransmitter. So really important for you to understand this process. So those neurotransmitters are released by the presynaptic neuron, bind to the postsynaptic neuron, and then might generate an action potential. And an action potential is this neural impulse which an electrical charge travels down the axon of a neuron, which causes the release of neurotransmitters. So we get that neurotransmitter release down in the terminal buttons of the axon. Important to understand that this is an all or none process. An action potential is either generated or not. Now, if you go to a more advanced neuroscience class, they're going to talk about excitatory inhibitory postsynaptic potentials. These are not action potentials. These are what lead to an action potential. So that's a very different process. But what happens is there is a critical point at which, once the cellular membrane has reached a particular point, that's when an action potential will be generated. So it's an all or none process. So once the synapse has enough neurotransmitter, the neuron will fire. That is, electrical signal will travel the length of the axon, cause the release of neurotransmitters, and the process moves on. Again, we refer to this level of neurotransmitter as the threshold. And once the threshold is passed, it doesn't matter how much more neurotransmitter there is, the neuron's just going to fire. Now, if after the neuron is fired and recovered, there is still enough neurotransmitter in the synapse, it will fire again. And so an important part of neurofunctioning is not just releasing those neurotransmitters, but turning them off or getting rid of them once the action potential has been generated. So that neurotransmission can be stopped by reuptake, by taking the neurotransmitter back into the presynaptic neuron, enzyme deactivation, which basically means there are enzymes in the synaptic cleft that deactivate or metabolize those substances, or what's called autoreception. And autoreception has to do with the postsynaptic neuron. So again, really great figure for you to spend some time looking at. Again, there are just plain PowerPoint presentations up on Blackboard for you to take a look at. So this is something you'll want to probably look at carefully. We have neurotransmitters that are synthesized in chemical building blocks called precursors. They're then stored in vesicles. And again, all of this is occurring in the cell body or somewhere along the way in the axon. Action potentials cause the vesicles then to fuse to the presynaptic membrane and release their contents into the synapse. The neurotransmitters are then released by and then binding to the postsynaptic receptors, and then neurotransmission is terminated by reuptake, enzyme deactivation, or autoreception. These autoreceptions here will shut off any release of neurotransmitter. One of the important parts to understanding how drugs function is understanding these different classes of neurons, because each neurotransmitter is going to be affected by different drugs differently. And drugs particularly are targeted towards specific neurotransmitter systems. So we're going to talk about acetylcholine, which is involved in memory and movement. It's one of the most basic neurotransmitters. Serotonin, which is involved in mood, sleep, and appetite. We talked a little bit already about GABA, which is gamma-aminobutyric acid, which is involved in sleep and is actually an inhibitory neurotransmitter. Glutamate, which is involved in memory formation. Norepinephrine, which is involved in arousal and mood. It's one of the adrenalins. Dopamine, involved in control of movement and sensations of pleasure. And then, of course, endorphins, which are involved in pain relief, and also some feelings of pleasure. So, in terms of neurotransmitters and their release, there are these 7 steps we've already talked about. Again, this is a separate review of the same figure we had just a moment ago. We have the synthesis of neurotransmitters, in which the neurotransmitters are formed or put together. There are precursors that are the main ingredient of those. They're brought to the neuron by the bloodstream. They're taken up by the cell body or the terminal buttons, and often come from substances in our diet. And in fact, we're probably familiar with some of these. For example, tryptophan, which is contained in a variety of foods like turkey, tends to affect the serotonin system. And so all those substances will come from our diet, and in fact are made from various proteins and enzymes. So these enzymes will then put all these ingredients together to create the neurotransmitter. The neurotransmitters, once they're created, are stored in vesicles, which are basically little small smaller cell bodies. They'll be concentrated and protected in those vesicles. They'll then be released through a process called exocytosis, which just means exiting the cell. So the vesicles will fuse with the presynaptic membrane and release their transmitters into the synapse. They'll then bind, in which they attach to in which the neurotransmitter attaches to a receptor. And again, they will attach to the postsynaptic receptor and presynaptic receptors. The presynaptic receptors will stop more vesicles from being released into the synapse. And so that's how we stop this process. As we talked previously, there are different varieties of receptors. Some respond very fast, and these are what we call ionotropic receptors. And there is a direct reaction to the presence of the transmitter in these ionotropic receptors. Remember when we talked about pharmacodynamics, talked about how some drugs will bind directly to the receptor, like a key in a lock, and cause that ion channel to open. So some neurotransmitter molecules bind to receptors on ion channels, and when a neurotransmitter molecule binds to an ionotropic receptor, the channel opens or closes, thereby altering the flow of ions into or out of the neuron. And so this is really critical in understanding how drugs work because they affect these ion channels. In fact, benzodiazepines work just like this. They affect these ion channels by affecting GABA. Other types of receptors respond more slowly, and they are indirectly affecting neurotransmission, and these are usually called metabotropic or G-protein coupled receptors. And what happens here is this initiates a secondary signal or messenger inside the neuron. So some neurotransmitter molecules bind to receptors on the membrane, signal proteins, which are linked to G proteins. When a neurotransmitter molecule binds to a metabotropic receptor, a subunit of the G protein breaks off into the neuron and either binds to an ion channel or stimulates the synthesis of a secondary messenger. And so rather than opening or closing an ion channel itself, it causes a later effect to open or close an ion channel. So once neurotransmission has occurred, again, we get inactivation by termination of the synaptic transmission through metabolism, reuptake, and reuptake by glial cells. And this only occurs for glutamate. And of course, we have autoreception shutting off the process at the presynaptic neuron. It's important to understand that there is a great deal of receptor specificity. Each neurotransmitter binds to several receptor subtypes. So, for example, the amines are a family or subfamily of neurotransmitters. The monoamines include catecholamines, which are norepinephrine, epinephrine, and dopamine. Indolamines, which is primarily serotonin. There are quaternary amines, which is acetylcholine. We've talked already about some of the amino acids involved in neurotransmission, like GABA, glutamate, and glycine. There are neuropeptides, which include the opioids. And then there are peptides, which include oxytocin, which you've probably heard about, is related to things like familial binding, infants binding or getting bound to their mothers emotionally, etc. Substance P, which is involved in pain, some of the hypothalamic releasing hormones, and then there are also gases that can affect neurotransmitters or function as neurotransmitters, like nitric oxide and carbon monoxide. So I want to first talk about serotonin. This is probably the one you've heard the most about. This regulates our sleep-wake cycle. It's also involved in regulating what we call homeostasis, which is keeping the body into its normal state, temperature, blood pressure, heart rate, keeping everything as it's supposed to be. So it's involved in mood regulation. It can also be involved in depression. Depression seems to result from diminished serotonin levels, although the current view of depression we'll see later on in the semester is it's more complicated than that. There's a dysregulation of serotonin levels. It's just not that there's lower levels of serotonin, that there is a dysfunction in the neurons, and by balancing out that system we can actually cure depression or at least treat it fairly effectively. And we talked earlier, or just a moment ago, about how turkey is related to tryptophan, which then can be metabolized into certain kinds of serotonin. So it's synthesized from tryptophan. There are a variety of receptors, both ionotropic and G-protein coupled. These are identified by the 5-HT1, 2, 3, and 4. When we get into talking about different types of drugs, we'll see that they affect these neurotransmitter systems, these receptors, differently. And in fact, some of them will bind only to 5-HT3 or 5-HT2, and it's important for us to understand that. Now, in terms of the pathways throughout the brain, serotonin largely is parallel to dopamine, and that's what the DA stands for. Dopamine is usually abbreviated as DA. And so, at this stage, don't focus too much on these 5-HT3, 1, 2, 4 things. I want you to focus on understanding how neurotransmitters work in the bigger picture. When we start talking about specific drugs, we'll get into these different receptors. Right now, it's just important for you to understand that there are different kinds of receptors. To give you an idea of how much serotonin there is in the brain or where it's used, as you can see, it seems to be everywhere. The serotonergic system is active in maintaining waking patterns. EEG refers to electroencephalography, which are brainwave patterns. Increases in serotonin activity are related to obsessive-compulsive disorder, tics, such as various tic disorders. Schizophrenia, certainly there is some serotonin dysregulation in schizophrenia. And decreases in serotonin activity are associated with depression. Now, again, not necessarily the cause, but are certainly a correlate of depression. So there are lots of serotonin systems throughout the brain. In fact, pretty much everywhere in the cortex. Our next neurotransmitter we'll talk about is dopamine. And dopamine is involved, again, in a variety of processes. Particularly, dopamine is involved in movement, learning, attention, and emotion, and in particular, our sense of reward. At a more basic level, dopamine is certainly associated with our ability to move, and in fact, our entire motor system is primarily regulated by dopamine. And in fact, Parkinson's disease is caused by disruption of dopamine-producing neurons in some subcortical structures we'll talk about later on. So Parkinson's disease is associated with relatively lower levels of dopamine, and in fact, the primary treatment for Parkinson's is giving patients a drug called L-Dopa, which increases dopamine in those structures. Schizophrenia is the exact opposite, and it's associated with relatively higher levels of dopamine. In fact, we know if we give Parkinson's patients too much dopamine, or L-Dopa, they can oftentimes develop schizophrenia symptoms. Similarly, drugs used to treat schizophrenia, which reduce levels of dopamine, can cause what's called pseudo-Parkinson's disease. And we're going to talk quite a bit about how antipsychotics can cause what are called extrapyramidal symptoms, which essentially is a pseudo-Parkinson's type of disease in which people tend to have movement disruption. So dopamine is really important for that particular system, the motor system. We also know that dopamine is involved in learning, attention, emotion, and what are called executive functions, working memory, our ability to make decisions. We'll talk about drugs like cocaine and how they affect decision-making capacity and reduce our ability to think about the consequences of actions, which is obviously important. There are three major dopamine systems throughout the brain. Lots of frontal cortex involvement in dopamine, and of course the basal ganglia, and the mesolimbocortical pathway and the mesostriatal pathways are involved in motor processes and motor learning, and then of course they have executive functions and all sorts of other functions as well. So the dopaminergic system is active in maintaining our normal behavior, and of course are related to Parkinson's and schizophrenia. You can see a variety of the different cortical structures that are involved in the dopaminergic system. Endorphins, which we talked briefly about a moment ago, are released in response to pain, high-stress situation, and also vigorous exercise, which is where the quote-unquote runner's high comes from. And in fact, morphine itself is what we call an endorphin agonist. That is, it helps these endorphins reduce pain response, which is why it's a pain reliever. And in fact, most of the pain relievers we're going to talk about are involved in endorphin systems, which is one of the reasons why they can be so addictive, because they do create that high that is associated with a great deal of endorphins. Big part of these endorphins are things like the norepinephrine system, or the noradrenergic system, which is active in maintaining emotional tone. Decreases in norepinephrine activity are related to depression, whereas increases in norepinephrine are thought to be related to mania. When we talk about bipolar disorder, that'll become important. Norepinephrine is an important stress hormone. In a fight or flight situation, norepinephrine would be released, and it causes a variety of behavioral consequences. When we talk about amphetamines, we'll talk about norepinephrine and how it's involved in the responses to amphetamines. And in fact, essentially what amphetamines do is they kick in our fight or flight system, which has some important consequences. Acetylcholine is actually the first neurotransmitter to be recognized because of its peripheral actions. It's involved in the peripheral nervous system, and in fact, it's also involved in motor activity, particularly in regulating sensory motor coordination and in getting muscles to fire. Acetylcholine is synthesized by acetyl-CoA in the mitochondria, which is of course involved in the Krebs cycle, and of course choline, which we get from our diet. So this is a pretty big process. In my entire life, I would never ask you to memorize this, but there it is if you're interested in it. Acetylcholine is inactivated by acetylcholine esterase, which we talked about previously. So after action in the postsynaptic cleft, acetylcholine esterase degrades acetylcholine to choline acetate, which are then taken back up into the neuron and then reused again in building more acetylcholine. Acetylcholine is produced in a variety of subcortical structures like the septal nucleus and the nucleus basalis, which include projections to the forebrain. These are part of the motor system. The midbrain, which includes the reticular formation, the pons, cerebellum, and cranial nerve nuclei. Again, great deal of involvement in motor coordination. The cerebellum is certainly responsible for coordinating our sensory motor activities. So this cholinergic system is active in maintaining our waking brainwave patterns, whereas serotonin is in our resting brainwave patterns. It's thought to play a role in memory by maintaining neuron excitability, and in fact, acetylcholine is one of the things that's affected a great deal by Alzheimer's disease, and a lot of the treatments for Alzheimer's is in boosting acetylcholine levels. The death of acetylcholine neurons and decreases in acetylcholine in the neocortex are thought to be related to Alzheimer's disease. There are a variety of receptors that are part of this acetylcholine system, including what are called the nicotinic receptors and the muscarinic receptors. And the nicotinic receptors are very important for how our memory functions, and nicotine, of course, affects those. There is actually what's called the NMDA receptor, which is the N-methyl-deaspartate receptor, which is very involved in nicotine, benzodiazepines, acetylcholine. Lots of different neurotransmitters are involved in that NMDA receptor. And we're going to talk more about those later on in the semester and how various drugs might affect these subreceptors. We talked a little bit about acetylcholine esterase inhibitors. Irreversible acetylcholine esterase inhibitors are generally toxic, and they are things like pesticides and nerve gases. And so these tend to be things that disrupt the acetylcholine process, will cause us to stop having motor functioning, which is why it's a pretty grim way to go. Also, a good reason to try to avoid pesticide exposure if you can make that happen. There are reversible acetylcholine esterase inhibitors, however, and these actually seem to function as cognitive enhancers and are shown to treat Alzheimer's disease, or are thought to treat Alzheimer's disease. So we also have the amino acids to talk about. These are synthesized from various other amino acids, like glutamine is synthesized from glutaminase, where we get glutamate, which is excitatory, and GABA, which is gamma-aminobutyric acid, which is inhibitory. More than half the synapses in the brain are affected by GABA. Glutamate comes from a metabolic pathway through the Krebs cycle, or from glutamate via glutamine, via glutaminase. Glutamate binds to several receptor types, including the NMDA receptor, kainate, and AMPA receptors. NMDA receptors are mediated by glutamate and glycine and serine. Again, these things will become important later. The NMDA receptor is a very important part of understanding how a variety of drugs work, and so we'll spend more time on it later. NMDA requires membrane depolarization by other substances, but we definitely know that the NMDA receptor is involved in memory formation. There are various disorders associated with disruptions in amino acids. Epilepsy and seizure disorders and dementias are all thought to be associated with disruptions in glutamate, and seizure disorders are probably associated with disruptions in GABA, because benzodiazepines, which affect GABA, seem to help treat epilepsy very well. So drugs that affect these different neurotransmitter systems that we'll talk about. There are some hallucinogenic drugs like PCP and ketamine, which are glutamate antagonists. And of course, we've already talked a little bit about some sedative hypnotics, such as benzodiazepines, tranquilizers, and also alcohol are all GABA agonists, and so they quiet the brain down. So this is actually a look at an NMDA